We have a question from the audience right here. Thank you.
This is a question for Dr. Reimer. So when you mentioned the healthier sources of fiber, like the pea fiber, and then they're put into a cookie, was there any aspect of the study that was done on, the presentation of the food? So then you get used to eating cookies, and if you're eating healthier cookies, that's okay. But what about the future?
Absolutely. So the form in which you add these ingredients can certainly influence what the subjects do during the study. We tried, within the context of the research, we try and control for that by also giving a cookie to those individuals that were in the control group. So they ate cookies as well, just like the ones that got the pea fiber enriched cookies did. But I think we're not necessarily trying to encourage individuals to eat more cookies every day. That's probably not the message. But I think the critical thing is that there's so many things we have put into our food supply that are not doing us benefits in terms of our health. And there's things we've taken out of foods that are harming our health. And one of the things that has come out of our food supply is dietary fiber. And so, hundreds of years ago, we ate up to 100 grams of fiber per day. And today, as Canadians, we eat about 14 grams. So we are well below the recommendations. And I think if there's a variety of foods that we can add the fiber back into, that's going to give us benefit, whatever the form may be. I have to say that the cookies tasted delicious. And the subjects who ate them for 3 months every day, they actually missed their cookies at the end of the study. So there might be some potential there.
Is there something special about pea fiber as opposed to other sources?
There's so many different types of dietary fibers, and they all seem to have a little bit of unique properties. They all share some common benefits, but typically there are benefits across the wide range of different dietary fibers. So we really do need a variety. If you just ate yellow pea fiber, that's not going to be the solution. So you need to have wheat bran fiber. You need to get it from a variety of sources to get the ultimate benefit in terms of health.
There's a question at the back. No, I'm calling you the back. It's actually, okay, halfway. Okay, let me count the...
Okay. So my question is prompted by what John said about cattle. We know more in Alberta about the heritage of cattle than we do about people because we don't have access to the data. But the question is really for Raylene, is what you said is for the generic person, which doesn't exist, right? Do we have enough data to distinguish categories and classes? For example, when we talked about the gut microbiome, right? There are 3 confirmed clusters of microbiome that cross nationality, countries, and everything. So we have that data, but we don't classify Albertans into those types because we don't get access to the data to be able to target what you've learned about fiber.
Right, and that is the future of medicine, and in the area, they call it nutrigenomics or nutrigenetics. So really matching up a person's genetic makeup and what their susceptibility is to disease or where they're protected against disease, and matching that up with the ideal diet. And it's not just diet in terms of lifestyle. What's going to work for them in terms of an exercise program? Does it have to be, they're going to have to be out running until they fall down every day, or could it be some weightlifting or whatnot? And so it's really matching up what is your genetic makeup, matching it with the ideal lifestyle intervention so that we can get the best benefits, so that you're not going to be a non-responder. We want to find who is out there that's going to be the responder to the particular intervention, because we don't want people wasting their time doing something, sacrificing something, battling through treatments that just may not give them any benefit. So, and same thing in terms of the gut microbiota. We have our own unique profile. Each of us has a fingerprint of our bacteria that is in our intestine. And so we can give you that profile very, very quickly today. But the key for moving health and moving medicine forward is let's now match up what your profile is to what should we do in terms of manipulating that, what's going to work best.
Maybe I'll add to that too as a clinician, because certainly right now we have to treat and we don't know which medication will give somebody this side effect or not, but we still have to do something. So you make your best guess, that's where the art comes in, but as we learn more about profiles and things, we hopefully can also streamline so you don't have to go through some treatment that's, gives side effects and things like that.
There's a hand up here. We need to get you a microphone. Oh, yeah, you have. Okay, go ahead.
I also had a question for Dr. Reimer. There's a very popular book out there called Wheat Belly, and yet you mention wheat as one of the sources of probiotic fiber. What's the situation?
Yes, so it's a very popular book, and I get asked a lot of questions about that because wheat is a staple in our North American diet, and we've eaten it for a very long time. And so I think for me, it always comes back to what's the evidence right now to refute or to actually agree with some of these books that come out into popular press. And we know that if an individual is not allergic to wheat, or they have the condition called celiac disease, when they absolutely have to cut out wheat or the gluten from their diet, that right now there isn't really any evidence that you're going to get all these health benefits from simply cutting wheat out of your diet. Why do some people get benefit when they remove the wheat from the diet? Well, it's likely because they've stopped eating things maybe they shouldn't have had too much of in the first place. So too much white bread and lots of bagels and pasta and, just too much of wheat products. So maybe that's why they see the benefits, not necessarily that the body couldn't handle the wheat. But that's a hot topic for debate, and it gets into crop manipulation too, because there's some indication that the wheat today isn't the wheat we ate 200 years ago. So there's many facets to that argument, and right now the bottom line seems to be that it is fine in terms of health to eat it, unless you are in those very defined health conditions where it's necessary to remove it.
And the truth is, if you write a book that is full of if or maybe not and qualifications, it won't sell. And so one always has to keep in the back of your mind that a book that is a strong piece of advocacy for something is probably leaving some data aside. There's a question? Yes.
Dr. Sim, is there any relationship between these prions and Alzheimer's? Like, is there any work going on in that area?
Did you plant that question?
Yeah. I was going to ask it.
That's an excellent question, and the short answer is yes. Alzheimer's disease and a number of other neurodegenerative diseases, so even Parkinson's, Huntington's, are associated with these proteins. In those cases, it's not prion protein, but a different protein, but it too changes shape and aggregates, sticks together, and causes damage to the brain. Similar. In addition, the way they spread through the brain may be very similar, and the diseases themselves are starting to be called prion-like or prianoid or prion-ish, or the terminology is changing. And what we are learning from research in prion disease is looking like it may have a lot of application to our understanding of these other more common diseases. So there are definitely some strong links, and there's a lot of active research trying to pursue those avenues and those links. And I think you'll hear a little bit about it later in a panel session with Dr. Cashman.
Yes.
My question's for Dr. Sim. In one of your slides, you showed infected brain tissue, and then you showed right beside it, with treatment, some recovery. Right. So what mechanisms are there for recovery, or what potential treatments?
Right, so that's an excellent, and I should clarify, so in that specific picture, there were two separate brain slices, so it wasn't the same slice that then recovered after treatment. So that's an important point, because what we do know about treatments in these diseases in particular, the earlier you start treatment, the better, and once you get past some point of no return, the likelihood of seeing that benefit can be lost. And there are several approaches to trying to treat these things. One is to target that misfolded shape itself. So you can either try to prevent the normal protein from becoming the wrong shape. You can try to take the one that is the wrong shape and get rid of it somehow, but it's hard to get rid of because it's very resistant to most methods. But try to prevent it from getting worse or getting bigger. So a lot of research has looked at trying to use what we call conversion inhibitors and blocking this. And while that sometimes works in the test tube, it doesn't work as well in the animal, and then the few that have made it into human trials, they really don't pan out. But again, the caveat there is by the time the treatment is started, often a lot of damage has been done. But another approach is to look at, well, what is the cascade of events in the cell that is triggered to cause the holes in the brain in the first place? So can we possibly target that shape shifting, keep that under control, but at the same time use a combination therapy and hit the point where those cells are starting to die and try to help them deal with the stress of all of this extra protein? And I think that's where a lot of people are looking now, is to try to combine those two. But it's difficult because just picking one particular treatment approach, it doesn't work. You say, oh, well, that's no good, and you try another one, and that doesn't work. I think we really need more than one target at the same time.
We do have so we have time for one more question, because the stunning thing is we're actually on time so far. And we're also, unbeknownst to all of you, monitoring the caffeine levels in your brain, and they're dropping. So we'll have one more question, and then we're going to have a 20-minute break for coffee. And if you have questions for any of the three speakers so far, you might be able to engage them at that time. So, yes.
Just a quick question for Dr. Sim. Has the Prion Center in Alberta, I heard that there was a possibility that funding has gone away. Are you still receiving funding for the Prion Center?
So there's a Prion Center in Edmonton. So we're called the Center for Prions and Protein Folding Diseases. Nice long acronym. There's also a prion group in Calgary. So they have a center as well where they can, it's smaller, but they can also do prion work. We have been basically supporting the center through the primary investigators. So each of us then seeks funding through our partners, through AI Bio and stuff, and through those funds are supporting the center itself. Certainly that's always a question because some funding comes through support of the university, some from the government, and of course a whole other topic for discussion is funding of universities right now. And universities, and U of A in particular, of course, has had a lot of those cutbacks. So certainly we, in addition to all of the other groups at the university, have fell on hard times with respect to funding, but we are still extremely active in writing grants and putting out the call for funding. If you're offering, we would love some. That would be great. But yeah, I think for all researchers in this climate, it is of concern. And I think these events are important to try to show you the purpose and the point of it, not just from the final applications, but right at the ground level, because without that entire continuum, you can't get to that end point. So.